Okay, hi everyone. Welcome to today's webinar. My name's Sam Jacobs from 6 Degrees Investor Relations, and I'll be hosting this presentation from Island Pharmaceuticals Managing Director, Dr. David Foster. In today's presentation, Dr. Foster will provide an overview of the top-line results from Island's Phase 2AB PROTECT trial using its lead drug candidate, ILA 101, in a human challenge model of dengue virus infection. And he'll also provide an update on the company's anticipated next steps around that and broader portfolio expansion initiatives. So as usual, after the presentation, we'll commence a Q&A. That'll be hosted by Dr. Foster alongside Island Chair Phil Lynch. That'll comprise both written submissions and a live question segment, which will be moderated during the webinar. Just lastly, a recording of the webinar will be available on Island's media channels later today. But yeah, for now, we'll commence the presentation. So David, the floor is yours.
That sounds great, Sam. Thank you very much. Thanks everyone for joining the webinar. I'm excited today to give you an update about ISLA 101 and the results from our PROTECT Phase 2 clinical trial. I'm going to get started right away. So Island Pharmaceuticals, just to set the stage, we are an antiviral drug development company. We like to develop small molecules, and we're looking for small molecules that come to us with a clinical history, evidence of a good safety profile that we can move into indications that we're interested in. Our lead indication is dengue infections. So there's about 400 million dengue infections a year. About 100 million of those require some form of intervention. And today there's no intervention. The standard of care for dengue infection is really fluids and pain medicine and rest. There's very little you can do about dengue today. It's a huge unmet medical need. As a result of that, there's major market opportunities. There's a huge potential market for a drug like ISLA 101. We're excited to be moving forward with ISLA 101. We came to today's announcement and discussion based on a lot of work done by some other people showing really good preclinical activity, which I'll tell you about as we get into the presentation. We've just concluded a Phase 2 clinical trial that I'm very excited about. I'm excited to tell you the results that we found there. And what this means is that we're going to get to move forward. We're going to get to continue with ISLA 101 to develop it for dengue infections. That creates a unique opportunity because of some of the characteristics around ISLA 101 that mean it's eligible for what's called a priority review voucher. Today, we check all the boxes to get a priority review voucher. And if that continues, upon approval, we would get that voucher. And the two most recent vouchers sold for around 225 million Aussie. So we think that's a huge opportunity. As many of you may be aware, we're also looking to pipeline expansion opportunities. I'll give you an update in the Q&A if anybody has any questions about that. I'm happy to talk about galidesivir and where we stand with that. Corporate overview, briefly, I'll run through this. We have about 236 million shares on issue. We closed yesterday at 16.5 cents, giving us a market cap of around $39 million. At the end of last quarter, we had $4.8 million in the bank. We have since raised another $3.6 million. So we're sitting in a very good financial situation at the bank. I'm grateful to our major shareholders. They've been longtime supporters of the company. And I just want to thank them for joining us on this journey as we move forward with ISLA 101. I'm proud to be working with Phil Lynch and Chris Timonopoulos. They are excellent directors, and I'm excited to see what we have in store, where we're going to be going next. So one of the core tenets of Island Pharmaceuticals is we really like the idea of repurposing drugs. To develop a drug from scratch is expensive. It takes a long time. It's around 10 to 15 years. It costs around $2 billion US to get a drug approved from start to finish. And frankly, we want no part of that. What we want to do is identify molecules that come to us with clinical history, a good safety profile, data where we can advance that in a unique indication that we're interested in. And that's exactly what we've done with ISLA 101. We're moving forward with a molecule that has a long clinical history, moving forward in a Phase 2 clinical trial. Phase 2 efficacy data is typically when you start to see significant value inflection points, when you see if your drug works in your indication of interest. And that's what we're focused on. I think this is an interesting slide. Not all indications are created equally. Many indications, there is a good um, correlation between preclinical work and the likelihood of getting that drug approved. As it turns out, infectious diseases ranks at the very top or close to the top of that list, where there's a greater likelihood of success based on preclinical models. You can see on this slide on the right about 15.5% of overall probability of success, which we think is very good in comparison to other indications like oncology, like CNS disorders. These are very low. The story actually gets a little bit better because, as you can see on the left side of the slide, once you have evidence of efficacy in a phase 2 study, the likelihood of advancing to a next phase 3 clinical trial increases dramatically, up to 64%. So we really like the idea that we de-risk the asset by getting a repurposed molecule in infectious diseases where there's already a greater likelihood of success. And after today, I hope you understand that we have very exciting phase 2 data, all of which suggests we're going to be moving forward with a subsequent clinical trial, getting us closer to approval and that priority review voucher that we're pretty excited about. Dengue, I've showed slides like this before. I like to show this map on the right. This shows dengue outbreaks over the last 3 months. And so it's an evolving map. It changes nearly every time I give this presentation. But I think if you take a look at Southeast Asia, if you look at India, if you look at Central and South America, it's clear they're absolutely on fire. Huge problems with dengue infections in these geographies. But interestingly, if you look in Northern Australia, we're also starting to see some outbreaks in Northern Australia, in Fiji. There's problems in southern part of the United States, as well as in California, which I find really surprising. So we're seeing the dengue virus and the mosquitoes that carry that virus move into geographies where you typically don't think about them. This is causing a significant problem as these new geographies are having to grapple with the problem of these viruses. The chart on the left, I think, is really important because it shows you 2024 was the worst year on record for dengue infections. A huge problem. There's a huge problem that's getting worse. And as of today, there's really nothing that can be done about it. We like to think that ISLA 101 could contribute to solving this problem or addressing this issue. Because the numbers are so large, I mentioned 400 million infections, 100 million of those need some form of intervention. The market potential is really staggering. So the dengue fever estimates for the market are around $2 billion today and increasing. And we think that's actually pretty realistic. Takeda is developing a vaccine. They've estimated the market for that to be around $1.6 billion. If you look at anti-malarials, which is a pretty close analog to what we're doing with ISLA 101, that's about a $1.7 billion market, and that's with inexpensive generic drugs. ISLA 101 would be a patent-protected drug that we think can make a huge dent in the problem for dengue infections. So ISLA 101, what is this molecule that I keep talking about? ISLA 101 is a fascinating molecule. It was initially developed by J&J about 30 years ago, and it was initially developed as a cancer drug. They developed it, they ran many clinical trials, demonstrated it has an excellent safety profile, but it didn't have great cancer activity. And so as a result, it's never been approved for anything. Nonetheless, many people continue to look at it. And again, just to reiterate, the drug has been in nearly 4,000 people in clinical trials. It's been in greater than 45 human clinical trials around the globe. But again, it's never been approved for anything. So we came to ISLA 101 because we were looking for molecules. Bill Garner and I were looking for molecules that had activity against flavivirus. So we were looking at Zika, we were looking at dengue and other mosquito-borne viruses, and we found some really elegant work out of Monash University. The investigators at Monash University, Professor David Jans and his colleagues, screened libraries of thousands of compounds looking for molecules that had anti-dengue activity, and they found one. They went on to show that it had activity against all 4 serotypes of dengue, as well as Zika, West Nile, yellow fever, and other viruses. They continued to show evidence of animal activity which I'll show you here in a slide or two. And importantly, about a month after the publication from Monash, Harvard published nearly the same work. They also screened libraries of thousands of compounds. And so you have these two world-class labs asking the same question, screening thousands of compounds, arriving at the same molecule, and then demonstrating the same activity. For me, the biochemist in me loves to see this when labs independently replicate each other's work. It's fantastic, and made us very excited to continue with the program. The one data slide I want to show you based on the preclinical work is here, and this is really the experiment that convinced me that I wanted to get involved with this program. I think it's been very important for a variety of other people who looked at the data and thought this is a very compelling package. What we're looking at here is an animal experiment. It's a mouse model of a lethal dengue infection. It's really a model of dengue hemorrhagic fever. And when you infect these mice with a strain of dengue 2, it's lethal. And so this experiment, we're looking at survival after infection in the presence or absence of ESIL 101. I draw your attention here to the red line. You can see after infection, by day 5, all of the control animals are dead. These are animals that were infected with no drug on board. However, if you give 1 dose a day of ESIL 101, we go to the green line here. And you can see about 20% of the animals survive. If we dose 2 doses a day, we go to here, the blue line. So about 70% of the animals survive. So in a dose-dependent fashion, ESIL 101 is preventing death in this otherwise lethal experiment. Very compelling data. Seeing the dose dependency really caught my eye. And again, made me think this is something I want to continue with. So we moved forward. We've completed a phase 1 study. We moved into a phase 2 clinical trial, which we call the PROTECT study. This is a challenge study that we ran in collaboration with the U.S. Army. We actually have a cooperative research and development agreement in place with the U.S. Army. And it's a challenge study in which we enroll healthy individuals, and then we infected them with an attenuated or a weakened strain of the dengue virus. People get sick, they get detectable viremia. It is a low-grade dengue infection. But the beautiful thing, because we control when these people are infected and it all takes place in a hospital setting, we can control when they get drug. We can either pre-treat them prior to infection and see if we can block the infection, or we can infect them first and then treat them afterwards and see once they're viremic, once they're showing symptoms, might we be able to help them to improve the symptoms? That's a challenge, but that's what we were looking at. That's what we wanted to do. So we set this trial up. The primary endpoint we were looking for is reduction in viremia. Secondary endpoints, we were looking at really signs and symptoms associated with the dengue infection. I put this in here just so that you could visually see the protocol that we were working with. So again, first we do the preventative cohort. We went to the FDA, we showed them this design. The FDA said, this is an excellent proof of concept experiment. We like that you're doing small number of individuals in the prevention cohort. But what we want you to do is after you do the prevention cohort, we want a team of experts, a safety review committee, to look at the data and answer, did you achieve the appropriate blood concentration of the drug? Was it safe? And did you see evidence of antiviral activity? And if the answer to all 3 of those questions is yes, then we're going to let you move forward to the treatment cohort. And so that's exactly what we did. We ran the preventative cohort. We had the safety review committee unblind the data. Only the safety review committee has ever seen the unblinded data until last week. And they concluded that they had seen evidence of antiviral activity, enabling us to move forward with the treatment cohort. And that's what we've recently unblinded, and we're going to tell you about today. So we have excellent activity. We're excited to tell you more. But again, this is all new to us. It's as new to us pretty much as it is to you. Before we get into our results, I want to set the stage for what the results from a dengue challenge infection look like. So this is published data. This is not our work, but it is an infection with the virus that we used in our clinical trial. I want to call out a couple of things. If you look at the blue trace right here, what we're looking at, this is the virus level, okay, viremia. And you can see there's this nice peak. The viremia goes up on about day 4. It peaks at about day 9. It's gone by about day 14. You can measure fever. This particular person didn't have a lot of fever, but this is what we're showing in red. We're measuring antibody responses to the virus. So this is a very powerful study. In addition, we're measuring symptoms. These are all symptoms associated with a dengue infection. This particular person talked about abdominal pain on about day 7. They had a headache on day 5. So this is what we're looking at when we set up to design our PROTECT study. So I know there's some lines on this, but I'm going to walk you through this. So the prophylactic study is pretty straightforward, right? We're going to dose before we infect. And so that's what we're showing here, dosing before we infect. There was a question around when do we start the treatment cohort? What we wanted to do, and this was very intentional, we wanted to start dosing the treatment cohort at a time that would be early in viremia, before peak viremia, and when these subjects were beginning to show symptoms. We didn't want them to be in full-blown symptoms, but beginning to show symptoms. What we were trying to do is mimic a wild type infection in an endemic country, because we wanted to really do an experiment to see if this would be effective in that setting. And so you can see here, we elected day 7 post-infection to start dosing. By this time in this individual, viremia is still on its way up. This person was complaining at this time. They were already symptomatic: abdominal pain, eye pain, fever, nausea and vomiting. So they were beginning to get sick. And we thought maybe there'll be a window within which we can capture a benefit here if we set up the study this way. So really, when we started the PROTECT study, the goal was very straightforward: to determine if ISLA 101 shows activity in humans against the dengue virus. Period. That's really what we wanted to know. If yes, we get to move forward. If it doesn't work, then the program would not proceed. So we set up this grid, really a go, no-go matrix. And if we saw positive activity in a preventative cohort or the therapeutic, or both preventative and therapeutic, then it's a go decision. However, if we don't see a signal, then that's the end of the line for this program. So we're obviously having a conversation. So what were the results? I'm incredibly happy to tell you we have very, very encouraging top-line results that strongly advocate for ongoing clinical development of ISLA 101 in dengue infections. In the preventative cohort, we see clear, meaningful reduction of viremia, viral load, and clear reduction of symptoms in the preventative cohort. Also, in the treatment cohort, we're seeing clear evidence of activity of the drug. We're seeing shorter viremia. We're seeing some improvements that we're still trying to work out exactly what these mean. And I'll show you what I'm talking about here in a minute. But I'm really happy about the results, because in this challenge model, which is a very rigorous challenge model, as I said, where people get viremic, they get sick. Other drugs have come through this particular model, and they've been unsuccessful. So we're actually the first molecule to go through the challenge model at SUNY Upstate and demonstrate a beneficial effect, to demonstrate that the individuals on our drug weren't as sick as the control. It's a huge moment. This is a monumental day for the company. And so let me walk you through some of the data that has gotten us very excited. So in fact, let me back up a little bit. On this curve right here, what I want to point out is one of the things we look at, and a lot of people would look at, is this viremia curve, okay? One of the results that we want to look at is whether we can demonstrate with ISLA 101 there is a reduction in the area under that curve. And we can do that a couple of ways. The area under the curve can be reduced by, one, reducing the peak viremia. If you don't get as much viremia, you have a reduced area under that curve. Likewise, we could reduce the duration of this infection, right? So there's a couple of parameters that we were going to look at to see how we affect the area under the curve. One of those is duration of viremia. So RNA-emia, which is the title of this slide, this is a measure of the virus. It's nothing more than that. It's a measure of the amount of virus in the blood of these individuals. And what we can see here is that in the placebo arm, there's detectable virus in the blood of these individuals for about 10.5 days. When we look to the treated arms, both the therapeutic and the prophylactic arm, as you can see here, show substantially reduced duration of RNA-emia or viremia. Rather than 10 and a half days, we're now down to about 8.5 days. So 2 days shorter duration, shorter exposure to the virus, which is a meaningful reduction. Again, in both of the arms. So we're very excited about this. I'm now going to walk you through a couple of other parameters. One, looking at peak viremia, which I just told you about, the peak, the maximum height of that curve, what are the values that we're seeing there? And then we're going to look at the area under the curve. On each of these graphs, you'll see black dots and red dots. These are just two different ways of measuring the RNA or the virus level. One is looking at the RNA or the nucleic acid that encodes the virus. The other, the red, is looking at active virus in the blood. I put both of these in here because they should track each other. They're really a control for the other. So I'm not going to spend too much time looking at the nuances between the difference, but I want you to pay attention to, they do track each other. There are trends when they go down, both of these values go down. So let's start with looking at viremia, looking at RNA-emia right here. In the placebo control, these individuals had, this is a log scale, so this is 10 to the 8th units of virus, if you will. This is about, we'll call it more than 10 million units of RNA. If you look at the therapeutic group, it's pretty much the same. Maybe it's gently lower, but it's not a lot lower. However, and again, each of these lines they're drawn across here, this is a 10-fold change, okay? So if you go to the prophylactic arm over here, this is a huge reduction in viremia. This is 10 to 15-fold less virus in the prophylactic setting. So pre-treatment results in about a 10 to 15-fold reduction of peak viremia. If you look at area under the curve, not surprisingly, we see this trend again downwards. So if we're looking at the placebo, we see around 60 units. If you look at the therapeutic group, there's a reduction, but the rather large error bars here in both of these, which I'll talk about shortly. And then in the prophylactic arm, again, I think a clear change from the placebo control to the prophylactic arm. So we have very strong signals of prophylactic activity. We're seeing trends of activity with the treatment cohort both of which are very exciting to us. This one I'm going to get into the weeds gently because I think it's incredibly cool, actually, what we're finding here. If we look at the peak viremia in two different ways, we showed you a collection of data from the group of the individuals in each cohort on the previous slide. In this slide, what we're showing you, each of these dots in this represents the viremia from one person in each of those cohorts. So if we look in the blue circles right here, this is my control, my three controls. We all know we have three controls. If we look in the pre-treatment, this is the purple line right here. These are my three pre-treated individuals. If we look in the red circles here, these are my treated individuals. I think I may have lost my pointer. These are the treated individuals. I think it's clear as between the control and the pre-treated individuals, clear reduction. Again, this is a 10 to 15-fold change in the level of virus. If we look over here, this is again measuring active virus, clear reduction between the control and the pre-treated individuals. But take a look what's happening here. There's a lot of my treated individuals that have low viremia. Likewise, there's a lot of my treated individuals with low viremia. And then there's this outlier. This outlier that frankly skews the numbers a little bit, caused us some confusion because they actually have elevated viremia and we were really confused about what this means. After doing some digging, we now know that particular person, it's the same person. This particular person has the lowest level of ESLA 101 of anybody in the study. And I think that's really important because what it does is it really highlights the fact that if you achieve the right blood concentration, we're seeing therapeutic and prophylactic effects. If you don't achieve the right blood concentration, then you don't get to control the virus. I think it's a really nice internal control, albeit one that confounded me for a little bit. So the take home here is in both of these cohorts, we're seeing evidence of reduced viremia. In the prophylactic cohort, it's a very strong signal. In the treatment cohort, it's a signal trending in the right direction, and we're still working to flesh this out. I want to change course a little bit now and talk about signs and symptoms. And I can see that this is probably a little bit fuzzy. I apologize for that. But like we did in the first slide showing you the dengue challenge, there's a variety of signs and symptoms that we're monitoring coming out of this study. And as it turns out, there's 11 signs and symptoms here. What we did was we looked at the number of individuals in each group. In the control group, there's 3. If there's 11 signs and symptoms, what that means is if everybody complained of every potential sign and symptom, that would be 100%. That would be 33 of 33 reported symptoms. As it turns out, if you count up the number of symptoms here, abdominal pain, 2 out of 3 complained, eye pain, 2 out of 3 complained, fatigue, all of them complained, fever, 2 out of 3. What you end up with is 21 out of 33 of the symptoms were reported by my control arm. Okay, that's about 64%. Now let's take a look at the prophylaxis data. Hop over here. Now what do you see? Abdominal pain, nobody complained. Bone pain, nobody. Eye pain, reduction from 2 to 33%. Fatigue, also a reduction. Fever, nobody complained of fever. As it turns out, when you count up the number of reported symptoms, it's only 11 out of the 33, so it's 33%. That's a reduction of about 50% of the reported symptoms between the control and the placebo arm, which I was thrilled to see. The fact that these people in the prophylactic cohort were better, they weren't as sick as the control arm. I think we've accomplished what we set out to do. We've demonstrated reduction in viremia. We've demonstrated reduction of signs and symptoms in the prophylactic cohort. I mean, it's a phenomenal result. If you look at the delayed treatment, there may be some hints here of reduced symptoms. It's not as clear, obviously, as with the prophylaxis. And when you count the number, it's about 61%, so pretty similar to the control. But we can dig into that a little bit more on the next slide. Here, we are looking at a couple of things. One, some of the symptoms from the previous slide. And we're also looking at lab abnormalities. So there are some traditional, typical lab abnormalities that show up in a dengue infection. And these would be when anybody goes to get a physical, they get blood work done, and they look at a lot of these, like your liver enzymes and things like that. That's what we're looking at. But if we go to the top here and look at some of these signs and symptoms, we see abdominal pain, fever, joint pain, and nausea. Again, 2/3 of the placebo group complained about these. But look at the prophylaxis group. Nobody complains of abdominal pain, no fever, very little joint pain, very little nausea. A huge signal here, which we're very excited about. If we go down here and look at the lab abnormalities, so now we're looking at leukopenia, thrombocytopenia. These are cell counts, ALT, AST. These are liver enzymes. Hypernatremia is looking at sodium levels in your blood. And what you find is that, again, there's a big signal with these people showing up with, they're not complaining of this. These are quantitative blood analyses that are being done. ALT, everybody complains of this or exhibits altered ALT. Thrombocytopenia, which is a reduction in platelets. That's really important for dengue infection. So 1 out of the 3 complained about that. But if you look at all treated individuals, this is prophylactic and treatment, nobody has thrombocytopenia in this study. Really interesting result, I think. There's a reduction in leukopenia. There's a reduction in all of these. And then again, if you look at the prophylaxis arm, many of these liver enzymes have the problems with the lab results, they resolved or substantially resolved. So a huge effect from signs and symptoms to lab abnormalities combined with reduction in viremia makes us, again, incredibly excited about what we've accomplished here. So what are the key findings and the next steps? Well, I hope you'll agree with me that the top line results demonstrate we have clearly positive signals providing evidence of activity from certainly the prophylactic arm, trends in the right direction in the treatment arm that we're going to continue to work on. So yes, is there a signal? Absolutely yes. We are going to be continuing with the program. The results from both arms are encouraging and advocate for additional clinical trials using ISLA 101. If we go to our go, no-go grid here, this bottom line, the no-go, we've eliminated that. We've seen activity. We're definitely moving forward. What do we do next? We're continuing to analyze data. We're working with regulatory advisors, clinical advisors, our scientific advisory board, a clinical advisory board that Phil Lynch and I met with in Singapore last week to pore through these data and get their feedback on what they saw. And they agreed we absolutely have a signal. We absolutely should move forward. And now we're going to be digesting this and putting together a plan for what the next study looks like. So again, two paths going forward. Yes, there's a strong suggestion that for moving forward with a prophylactic study next. There's some hints of evidence that we should move forward with the treatment. I think it's today, I would say that there's a stronger signal we should move forward with the prophylactic, but that really remains to be determined after we have continued conversations with our experts and get additional feedback. But I think Stephen Thomas, who sits on our scientific advisory board, had a really nice comment. And he pointed to there's 400 million infections. There's very little in the clinic that is being developed for dengue treatments. And so for this reason, we're very excited. Island's candidate, ISLA 101, demonstrated evidence of antiviral activity against a rigorous dengue human infection model. These results set the stage for continued development. And that's exactly what we're going to do. So we're going to walk away from this. We're going to continue reviewing the data. We're going to be talking with our experts and putting together a plan, and we'll keep you across it as soon as we possibly can. So with that, I'm going to pause. I'm going to stop sharing my screen, and then I'll be available for questions.
Thank you, Dr. Foster. That's much appreciated. And we've got quite a few questions here that have come in from the floor. So we'll work through these with David alongside Phil Lynch. David, perhaps just to kick off the Q&A, this is, quite a simple question, but it's a variation on a theme that came through from a few investors. Ultimately, how close were these results, do you think, to what you expected going in?
Yeah, so that's a really interesting question. Going into this study, we had seen the robust preclinical animal data that I told you about. We knew that there was a higher likelihood of success in infectious diseases compared to other indications. So we were confident going into this that we would see a signal. And that was certainly our hypothesis when we started this, that we would see a signal. I thought there was a greater likelihood of success with the prophylactic arm compared to the therapeutic arm. But let me also throw the caveat in there is there's other drugs that have gone through animal models and other challenge studies. They had a right to think they were going to be successful, and they weren't. And so while our hypothesis was that that it would work, I'm thrilled. I think we're all thrilled that it worked, that we see the clear signal that we see. So yeah, we're incredibly happy with the results.
Yeah, fantastic. And just sort of on the same theme, but perhaps just with reference to what you discussed in slide 23 there with how this data is going to be analysed in conjunction with the advisory board. Do you think there's going to be additional data from that process that the market can expect to see?
Yeah, so I think the very important top-line data, viremia signs and symptoms, you've seen it. We've just showed you a lot of what we have with respect to that. Having said that, there's a lot of exploratory endpoints looking at antibody responses, immune response to virus, looking at cytokines. There's a whole slew of things that we're studying right now. We will announce those to the extent they're relevant as we get them. Likely some of this will get published in scientific journals over time, but yeah, to the extent it's relevant to the advancement of this program, of course, we'll keep you across it. There's going to be plenty more data coming out of this.
Yeah, fantastic. And just with reference to those potential pathways, whether it's prophylactic or therapeutic, we've had a question come in from the floor in connection with the priority review voucher process. So do you think, a decision to progress with either of those pathways will have any bearing on the potential for the company to secure a priority review voucher, or its potential value to a third party?
Yeah, so that's a really good question because they're different, right? A prophylactic and a treatment, they're different. In order to get priority review, you have to check three boxes basically. Your drug has to get priority review itself. The drug has to be first approved in an indication on a list of eligible indications. So currently, we believe, because there's really nothing that can be done against dengue and severe dengue at this point, we believe that as a therapeutic or a prophylactic, we would tick that box, that we ought to be eligible to get priority review ourselves. ISO 101 is not approved. Dengue's on the list of diseases for which you can get a PRV. So yeah, we believe we tick all those boxes today, whether it's a prophylactic or a treatment.
Yeah, fantastic. Thanks, Deb. So we've just had another question come in. It's probably more a simple line of questioning, but just for the retail investor base, one investor just asked, can you please give a quick explainer just for the uninitiated, what viremia actually is? My understanding is connection with that viral load, but is there a quick explainer you can just describe that for us?
Yeah, absolutely. So viremia is a measure of the number of viral particles that are circulating in your blood. Increased viremia means you have more virus. Reduced viremia, you have less virus. When you get your first infection, you typically have low viremia, and then the virus propagates and multiplies, and then your viremia increases. So it's really just a measure of the level of virus in your blood.
Nice one. Thanks, David. So yeah, there's quite a few questions coming in connection with that FDA, broader FDA approval process. So perhaps can you talk to, what are the key data points that the FDA look for this trial, with these kind of results?
Yeah, of course. So the FDA's primary concern always is safety, right? We want to make sure that we're giving people a safe drug. We didn't see any safety signals in this study, which we didn't expect to because the drug has a well-known safety profile. But that's always going to be what they're looking at. The primary endpoint was a reduction in viremia. So we have hit that endpoint. I think that's very clear that we will be able to demonstrate that. They're really going to look to the endpoints that we have told them we want to meet and see if we met them. And I think in this situation, they will be able to say we've met the primary endpoint of reduced viremia. We've also met the secondary endpoint of improvement in signs and symptoms. And that's really what they're going to be looking for.
Gotcha. Thank you. A questions come through just with reference to slide 22 or 23, I think it is, just on key findings and next steps. One investor just asked, with the reference to potentially positive for the therapeutic line, with reference to the 2B study design, could there have been a scenario where that therapeutic data would have allowed you to conclude, directly positive rather than potentially positive, or was that just the relevant signpost given, this current stage of the trial design?
Yeah, so we always thought the therapeutic arm was going to be a heavy lift to see a robust signal. Had we seen, a robust signal, more robust than what we saw? I mean, we've seen nuanced effects on signs and symptoms. We've seen a reduction in viremia, not as robust as prophylactic. It's hard for me really to imagine something being a lot better than that. We didn't expect, for instance, viremia just to drop straight down once we dose drug, right? We thought, if anything, we might be able to tease apart an enhanced rate of decline of viremia. And these are things we're actually still looking at. But those are pretty nuanced. So, the fact that we're seeing shorter duration of viremia, I think that's a great signal. That we're seeing some reduction in area under the curve, with the caveat that there's some pretty big error bars, which I think we can now explain based on that one outlier person that we observed. So I think the therapeutic results, that might be what we should have expected to get.
Yeah, that's interesting. Thanks, David. There's now quite a few questions on the sort of next steps in the pathway. So turning from the technical aspects of the trial itself, to what's next. One question was, is the company considering partnering for its next trial initiative to broadly reduce costs?
Yeah, so partnering is a really interesting part of the strategy. And it's, if you've heard me talk over the last couple of years, you'll know that I've been to partnering conferences all over the world, really planting the seeds with companies in relevant jurisdictions like Singapore, like Brazil, letting them know that should be on their radar, that we're going to be running this study. And once we have results, we want to continue the conversation with them. So there's a number of companies with whom we are talking right now based on these results. Chris Tumanopoulos and I were just in Brazil and had phenomenal meetings with a bunch of companies that focus on Latin America health concerns, dengue being a primary one. So we think that as we look to doing clinical trials in different geographies, it probably makes sense to think about partnering. And so, yeah, that's certainly a part of the strategy that we think about.
Yeah, well noted. And perhaps in connection with that, yeah, just looking, given the company's communications to date, would the company consider running a trial potentially in different jurisdictions? I think with reference to the last fundraising announcement, some of those funds were earmarked to trials in key international markets. Yeah, obviously where dengue is more prevalent.
Yeah, so that's really the key, right? Is if we're going to do a field study next, we're going to a field with endemic disease. And so think Southeast Asia, think Latin America, think India potentially. These are all geographies that have big dengue problems. They have expertise, dengue expertise in those regions. That would make a lot of sense for us to be thinking about running a study in one or more of those regions.
Yeah, fantastic. There's just turning back to the trial itself, because we've had a couple of attendees ask about the slide with reference to where you were talking through that reduction in viral load, those interesting charts there. And I think you covered off on this, David, in some detail, but we've just had quite a few attendees who are interested on this point. So perhaps if we can just go back over talking through why those outliers popped up in the data and the explanation for that in connection with the potential use case for ISLA 101.
Yeah, so the outlier that I think people are asking about is in the treatment cohort. We saw there were clear signs of reduced viremia in the treatment cohort, yet there was also an outlier that had actually increased viremia compared to control. And that really confounded me. We had a lot of discussion around that, and in fact, went back to look at that individual and find out what was going on with them. And as it turns out, they happened to, for some reason that I don't know yet, they had the lowest level of ISLA 101 of anybody in the study. So, my thinking on this is that, maybe they didn't take the drug with the appropriate diet. They didn't take all the pills they were supposed to. I'm not entirely sure yet, but we have that information's available once I dig it out. But the important thing is that it shows that if you don't achieve a certain blood concentration, then the drug may not be effective, which is not a surprise. We were always targeting a specific drug concentration, and for the most part, we hit it, aside from this one person.
Yeah, that's well noted. Thanks for covering back off on that, David. We should probably turn to, because this question will probably represent a number of questions from the floor, but just in terms of turning from the trial to the broader asset portfolio, are there some updates you can provide on discussions with BioCryst and where that's at?
Yeah. So BioCryst is a company we've been talking with for well over a year. They have a really interesting molecule called galidesivir. We are big fans of galidesivir for a variety of reasons, including it has great activity. There's some interesting regulatory clinical paths that you've probably heard me talk about before. And if not, I'm happy to have that discussion maybe another day, but it's a really fascinating molecule. Maybe around 10 months ago or 9 months ago, we executed a binding letter of intent for an option to acquire that asset. And since then, we have continued to do substantial due diligence on that asset. We, in parallel, have been talking with BioCryst to finalize documents associated with that transaction. The pace of those conversations is increasing. Things look good. We are really hopeful to have an update in the not too distant future. I can't make any promises as to timing because I'm not the only one who negotiates that or prepares those documents. But we're working as fast as we can, and we hope to give an update in the not too distant future. That's probably the safest thing I could say.
Yeah, fair enough too. Thanks, David. Yeah, turning back to the treatment, based on the potential approval pathway for the prophylactic, that preventative measure for ISLA 101, can you talk about what you think that potential use case, or just give some extra context around what target market you look at and the potential size of that market for that pathway?
Yeah. So as if we're going to consider prophylactic, so pre-treating people before they're exposed, there's a variety of ways I think about this. One is obviously travelers, people traveling to endemic countries who are concerned about this. Again, we see this play out. This is why I draw the analogy to the anti-malarials, because that's really a lot of what they do as a traveler's market. Likewise, we think the military in the US and around the globe could be very interested as they're sending soldiers into endemic countries. I mentioned at the outset that we ran this study in collaboration with the US Army. We know that they're aware of what we're doing. We have a CRADA, Cooperative Research and Development Agreement, in place with them. So we know that they're keen to be following what we're doing. So I think that makes a lot of sense. The other thing that these data really suggest to me is there may be an opportunity to capture what I would call a post-exposure prophylactic opportunity. And that's distinct from treatment. So what I would call post-exposure prophylactic would be you've been infected, you don't know it, you're not exhibiting symptoms yet. And if you were to take the drug at that point in time, I think that there may be a window, and the data that we presented today actually support this. There may be a window to see some therapeutic benefits, maybe reduced viremia, maybe reduced signs and symptoms. And the way that works, in my mind at least, is in a lot of these countries with endemic dengue, these are really crowded populations. They're crowded homes, they're crowded hospitals. And in fact, when I was in Brazil, had a conversation with a guy who had just had dengue, and we were talking about ISLA 101. And he said, if I had this molecule, not only would I have wanted to take it, I would have wanted to put my entire family on it, even if I didn't know they were infected. And I think that was a really important statement that he made, because what it suggests to me is that people really would want to protect anybody else from getting this, even if they didn't know whether or not they had been infected, if they weren't having symptoms yet. And that is, I think, a really unique opportunity that we may have. It's going to take us some work to get there, but I see this as prophylactic, travelers, military, potential post-exposure prophylactic.
Yeah, that's really interesting. So I think just staying with the trial results for a second, there's quite a bit of interest around the idea of kind of comparative trials. So perhaps, taking into account you might be limited on how much you could talk to other trials, but one question that kind of represents this general interest from the floor is how do prophylactic responses compare, do you think, to the prophylactic phase 2 trials and challenge studies, for example, the Takeda vaccine? Is that sort of a relevant base point for you to kind of discuss that with reference to other research in the market?
So, the vaccines are unique, and we can talk about them. Those, they're large clinical trials. They're really sort of come all clinical trials to a population of people in an endemic area. And as a result, they can get big. One of the interesting things about the vaccines that are in development is well, let me preface this by saying, in order to develop the optimal dengue vaccine, what you need is a vaccine that has activity against all 4 serotypes at roughly equal efficacy, probably greater than 90% efficacy. And that vaccine doesn't exist. Okay. That's just not out there. The Takeda vaccine is arguably the best one out there right now. And they're doing really good things with it. I applaud them for the work that they're doing. But those clinical trials are large because they're really looking at the effect on all of these different viruses, all of the different serotypes of viruses in a general population. One of the things that we think about is, might there be ways to enrich a study for people who might be at risk for more severe dengue, for instance. I think that might be a way that we could reduce the sample size that we might need to look at. We might see a bigger signal. The size of a clinical trial is really determined by the size of the signal you're looking to observe. And so we think if we can find a situation where we can enroll people who may be at risk, where we could see a bigger delta between control and treated, that might shrink the numbers. And we're looking into that.
Yeah, interesting. That probably connects to this next question. I think you've covered off on a broad spectrum of topics through that Q&A. So perhaps to round it out with more of a forward-looking question, yeah, are there some points you can talk to? I mean, various attendees have asked about kind of what's next in the program, with obvious reference to what you flagged in the presentation. But some of those attendee questions include, are preparations underway for a phase 3 trial? Are you preparing to potentially increase dosing levels? So is there sort of a general wrap you can give us here for this Q&A in terms of how you're thinking about the next step of the trial, obviously in conjunction with your advisory board going forward?
Yeah. Yeah, I'll tell you what I can. I think it's not going to be a phase 3 study that we do next. It would be another phase 2 efficacy study looking probably at smaller numbers. We'd like to see it follow the trend of seeing the signals that we're observing right now. I'm going to, without being certain, I think the trend is probably that prophylactic is the way to go based on the data that I've shown you today. Although treatment still could be explored, and we're going to be looking at that. So I have to be really careful and say the jury's still out. We're working with our advisors. We're going through data. But that's kind of the way I think about it. With respect to increased dosing, that's a possibility. I mean, we're happy with the dose that we achieved, the blood concentration we achieved, but we can certainly explore that. And we can do that, based in animal studies. We can look and find ways to do that. So there's a variety of things we can think about. Yeah.
Yeah, fantastic. So I think there's quite a bit for investors to look forward to there. And thanks, David, for that comprehensive Q&A in addition to the presentation. I think we'll wrap it up there. But our obvious thanks to all those attendees as well, and all the questions from the floor. There was a lot of interest in the end, so apologies there if we didn't get to everyone's questions. We tried to sort of group questions where we could. So yeah, look, as mentioned earlier, a recording of this webinar will be available to come back to on Island's website, associated media platforms. That should be up later today. But just given the presentation and that Q&A, David, just before we go, are there any final points that you or Phil would like to add?
I'll start, Phil, and turn it over to you. I want to thank people for coming on the journey with us. It has been an exciting journey, and I hope that today you're satisfied that we've achieved an exciting result. We're certainly excited. We're moving forward. We've got opportunities in front of us to make a big difference. So I'm available to answer any questions. Feel free to reach out to me. Phil, obviously want to thank you for your support and all the work. Phil and I were just in Singapore having a great meeting with our advisors and attending an invitation-only dengue conference, which we were happy to be at. But Phil, I'll turn it over to you.
Yeah, thanks, David. And I think you've done a great job at giving everybody a good update of what we know so far, but there's a lot more to learn. I would simply say that this remains a significant unmet healthcare need. We've had historically, as David said, good data. We've now got continued good data that gives us confidence that there is an economic opportunity, a healthcare opportunity, but we need to be guided by how can we best deliver that in a way that is meaningful, that in a way that's affordable, and it needs to make an impact in the broader healthcare community. That's not an easy question to answer, which is why we're somewhat saying, and I'll say again, we will defer to expertise and we'll continue to consult with expertise. The type of trials we may contemplate and proceed with are expensive. They're investments and they need to be well considered. So you'll hear that from us. We won't commit to a date to say when we'll give you that update, but rest assured we're working very hard on it. And David, I want to commend you for the journey that you've been on over 5 years now, and I know you're excited and very committed to see us landing this.
Absolutely, Phil. Thanks.
Yeah, thanks, Phil. And thanks again, David. Look, I think that wraps things up for now. So thanks again to the presenters and all of our attendees. We'll see you next time.
Very good. Thanks, everyone. Cheers.
Thanks, everyone. Bye.